Okay, thank you everybody for coming. So this is the last meeting of the AI Club for 2024. So I am happy to introduce Arun. So I've known him for a long time because I worked with him while he was completing his PhD in infectious diseases. And today he's going to talk about a topic that is very interesting to me. So I teach a course called computational biology, and one of the things we talk about in there is the prediction of protein structure. And computer scientists are really interested in this problem for a variety of reasons. But one of them was that it was suspected that the first person to truly solve this problem might actually end up winning a Nobel Prize. And we've thought this for 20, 25 years. As computer scientists, we typically don't have the opportunity to win a Nobel Prize. There's something called the Turing Award, which is kind of like the Nobel Prize of computer science. But this is a scientific problem, protein structure prediction. And it turns out that came true. Actually, the people that solved the problem of protein structure prediction solved it so thoroughly that it really blew all of the other methods out of the water and did indeed win a Nobel Prize for it. So that's what Arun's going to talk about today. So, like I said, Arun earned his PhD here in infectious diseases, and then he went on to do his postdoc at Woods Hole Oceanographic Institute, and then he returned here, and he is a research professor at the Global Center for Aquatic Health and Food Security. So he's going to talk today about the journey of AI's first Nobel Prize by unraveling the 3D protein structure. Arun, take it away.
Thank you. Thank you, Dr. Perkins. I still remember coming to classes here. I started my journey with the computer science, taking Dr. Perkins' computational biology and bioinformatics. That's where I started developing my interest in this field. So my major, as he mentioned, is infectious disease, mainly with viruses. So I'm a virologist. So my PhD, I started in College of Veterinary Medicine in 2017. But towards the end of that, I got more interested into computational biology. And finally, I ended up taking a minor in computer science also, along with my major in infectious disease. Interestingly, this AlphaFold 2, which is the program which solved this problem, I actually used that during my PhD. So today we would look at some little bit of history. And as an introduction, this is not a new problem. It's been there for around 40 to 50 years. Different people, the most smartest people in the world, tried different ways to solve this problem. And finally, because it's very complicated, it can get very hard. So finally, it is like, because there are so many moving components in that, Google's DeepMind got it right. So, and it did got a Nobel Prize for that. But just about me, I'm actually works in Gulf Coast Aquatic Animal Health Lab in the Gulf Port. So my main work is to work with genomic epidemiology, One Health, bioinformatics, high performance computing, and molecular biology. So this is my areas. So we are just starting the lab. So that's about my background. Now, even though the title says 2024 Nobel Prize, that's for AI. However, this problem actually got another Nobel Prize in 1972. So structure prediction problem actually got 2 Nobel Prize, and you can see the gap, 1972 and 2024. So in 1972, first part got solved. Now, it took this many years of work, and finally, AI was capable of solving that problem. And for that, David Baker, Demis Hassabis, and John Jumper, they got the Nobel Prize. And he actually works on the design of the protein, and these two are basically from Google's DeepMind, and they are the computational biologists whose idea behind solving this problem using deep learning. So we will see how it progressed. So first, as a background, what are we dealing with? What is protein? That is the first question, very, very, very important question. Proteins are nothing but biomolecules. So in your body, there are so many proteins. Most common example, our hair, it's keratin, it's a protein. So our body needs different types of proteins to do day-to-day functions. It's not only in our body, viruses has it, bacteria has it, any living organism has proteins, and they are actually the workhorses which gets things done. So they are the most complex, one of the most important molecules in the living organisms when we really look into the details. But before getting into that, there is a very basic concept which is called central dogma. So which says DNA to RNA to protein. Let me tell you what it is. So if anyone doesn't know what is DNA, DNA is the molecule which stores all our genetic information. So if someone sequences my DNA, it tells about everything about me. It tells about my parents, my genetic lineage. Probably you might have heard 21andMe. You send or heredity. You send your thing so you can find out about your ancestors. So DNA is kind of the secret of the code, secret of the life, where all your informations are stored, and basically it also encodes all the recipes. So I always consider it as a recipe book. Every page has a recipe for one thing. So for example, we got a haircut. The hair has to regrow. Then our body needs to know what is the recipe of the hair. So there is a code for that. So this is a very long molecule which encodes the recipe or the secrets for anything, everything like happening in your body. Now, the DNA is actually made up of just 4 molecules, A, T, G, C. So they are adenine or guanine. They are chemical structures. These 4. So for simplicity, we always make it as 4 letters, A, T, G, C. It's their combination. As their combination changes, what information is stored also changes. Now, when, say, again, coming back to the other example, say our hair has to grow, our body asks, then DNA actually sends a message. So for example, if this is the part which needs, it makes a copy of that. So that's what is this mRNA, which is basically messenger. So it makes a copy and give to another part of the cell, hey, here is the recipe. Now, inside the cell at some place, based on this message, it makes protein. So protein is actually, so you can actually see this is DNA, this is RNA, which is the message or the particular part which we are interested, which the cell has to make. Then it makes a protein, which is a 3-dimensional structure. So even though it looks like this, you can actually maybe imagine like a football. It's a 3-dimensional structure. The most key factor we need to remember is, now we saw the sequence, okay? These are all like, let's consider string. It's a string pattern. But ultimately in the body, what matters is this 3-dimensional structure. So whatever happens here, it's all fine. But at the end of the day, if you can make this structure properly, then your function would work. So it could be immune system. You have to fight against the disease. It could be your digestive enzymes. So anything. So basically, it's the structures. Now, coming to the protein structures, what is the building blocks of protein? It's called amino acid. Again, they are chemicals biomolecules, there is 20 naturally occurring amino acid. Now, whatever which has to happen or has to make in your body, it's just the combination of this 20 amino acids. So they are the building blocks. So some proteins can be only 100 amino acids. There are some proteins we will see later, it has 1100 or 1200 amino acids. So the amino acids always serves as the building block to protein. They form a bond called peptide bond. So then one interesting thing is then they actually form a two-dimensional structure and they form a three-dimensional structure. So one thing we need to remember about the amino acid, there are different types of amino acid. Some of them has charge, positive charge, some of them negative charge. Now, very simple, positive charge, repel. Positive and negative attracts. And some of them are hydrophobic, means they don't like the water. Some of them are hydrophilic, they love water. So what I'm trying to say, there are physical properties which defines each amino acid. So you put 50 of that based on what charge, or is it hydrophilic, they just interact each other, they form this, and finally they just form this. So it's a physical process in some context. Based on its geometry and based on its physical properties, they just interact and they just form a three-dimensional structure. Now, with the technology, we already know the DNA sequence. However, if we know the DNA sequence or an amino acid, say there is 100 amino acids, we know 100 amino acids, then we want to know what would be this 3D structure. So that's a problem. This is a problem which was haunting everyone for 50 years. We do know this, but we were not able to figure this out. It's already there. So, of course, there is experimental method, which is called X-ray crystallography. You grow the crystal, you use a diffraction technique. You can get this structure. But the problem is it takes years and years to do this. And some proteins, it doesn't work and it's very expensive. So we need answers. Of course, then we want to do the modeling, because if we can do in silico method without going to the experiments, which can considerably save our time, effort, and cost. So that is the whole point of people wants to solve this problem. So there are different ways to predict this three-dimensional structure. And before getting into that, there is actually, so when the amino acid interacts, it actually forms two type of blocks. One is called alpha helix, one is beta pleated sheet. So they are just like the helix or like a cylinder. This is nothing but like a sheet. Now, what happens is, first Nobel Prize, the 1972, he was the first one figured out that this 3D structure really depends on what is the combination of this. So if this sequence changes, this would change. I'm making it too simple. However, that was also a hard problem. For that only, in 1972, the first Nobel Prize was awarded, saying that 3D structure of the protein depends on the amino acid combination. It's as simple as that. Now, from there, so we know it's the structures. So the question is, okay, we know the combination. Can we predict the structure? Again, there is two type of method for this. One is template-based. Template-based, experimentally determined structures are there. We know these are the structures, and we try to learn from that and use the modeling to predict a new structure. And there is actually, it's called ab initio, which is totally physics-based. That's what I said, positive charge, negative charge, all the geometry and physical constraints it's used. So it's physics-based, ab initio. The ab initio of the problem is, it's like when we think it's exponentially gets complicated, and sometimes it doesn't work either. Now, after 1972, there is two things happening parallel. One is this protein structure. Now, you all might already know the AI's progress too. So slowly you can see in 1990s, 95, deep learning started. Then eventually you already might know AlphaGo happened, where it actually beat the Go problem. That's where really the deep learning and CNN, all the neural networks really picked up. On parallel, this protein structure problem was also going on. So two fields independently which was developing all this time. Now, during 90s, what happened is everyone wants to do the modeling. Everyone started predicting the structure. Now, as you all know, what happens with the modeling? It's just a model. How do we know it's right or wrong? And people really went on that. What happened is you just overtrained almost all the models, and usually it doesn't reflect what is the actual structures. So there is some people said, oh, no, we cannot keep on generating the structures like that. We cannot do the modeling like that. We need to know how to validate this. So they actually started a competition. It's called critical assessment of structure prediction. It's a gold standard test. It's very simple. Someone actually determined the experimental structure, but they will not release the data. Then we call all the computers, hey, bring all your models, give them that question. However, we already have the experimental structure, even though the modeling people does not know. Then what they do, they compare the models along with the actual structure. It didn't go well. People really didn't like it. The standards were very high, and it actually made some people leaving the field because it really doesn't worth it, because it's getting tougher and tougher. However, finally, then, as I said, as the deep learning really picked up, then Google's DeepMind, they decided to work on this problem. So it was going on, but they really started putting resources on this. Now, yeah.
There is one glitch actually. The desktop is not being shared.
Oh, okay.
Just 1 second.
For the
Recording.
okay. Oh, okay. So finally, how they did it. So the program is called AlphaFold. They had a first version, which was okay. The breakthrough happened in AlphaFold 2, which is came out during 2019 or 2020. Now, as you can see what they did, they combined the AI capabilities, they combined the evolution, and they combined the physics, chemistry, and existing template. So that is what really changed the picture, because they were able to bring all those together. So they got it right. Now, as for AlphaFold 2, this is the entire network. So we'll look at some in some details. So this is called AlphaFold 2. It has basically two components. One is inputs. So what is the input data? Second is what are the networks? And finally, how you get the results. Let me see what is so for inputs, we have actually mainly three things. One is called multiple sequence alignment. Second is the pairwise data. Third is the template. Now let me explain what are these. So multiple sequence alignment, it's simple. I told you we have the amino acid sequence, right? So if amino acid sequence from human, there is fish or chicken, we want to compare that. So that is basically multiple sequences. So those amino acid sequences are aligned to see the to compare those. So that is multiple sequence alignment, and which one more second.
Yeah, I'm so sorry. I don't know why it's not working.
Oh.
It should work, but
Easy.
The system is like crashing.
Okay. So multiple sequence alignment. Is that working? So what is the importance of multiple sequence alignment? It basically tells you the evolution. For example, you take a protein from fish, chicken, and human. If you basically align one by one, you take the position one, you say, oh, okay, this is the amino acid. It's same for fish, human, and chicken. But on second position, in humans has a different amino acid, but chicken and fish has same. So it basically tells how things evolve. That's the information we get from MSA. So if you have a sequence, there is actually genetic databases already has this information, and it creates that. Now the second one is pairing. Pairing is simple. So imagine you have 50 amino acid, 1, 2, 3, 4, up to 50. It simply makes pairs like 1 to 2, 1 to 3, 1 to 4, 1 to 5. It's a pair matrix, nothing special about that. So it's basically pairs those. You can see that. Then it has another basically a supporting structure database. Those are the structures which is already experimentally determined. So we have multiple sequence alignment, we have a pair data, and we also have structural database. So these are the inputs. Then your neural networks, different components of that, the final structure. So let's get into some of this detail. So again, coming into the input, little bit detail. So this is what is residue pairs. I just told what is pairing. So you can clearly see that they are simply pairing the blue and the red. You can see. So it's just the pair data. Now, so what is important? Yeah, that's in the next slide. Okay. So the multiple sequence alignment tells you about, I told you, the evolution and the function. The conserved variable. Okay, let me try to explain this. So there's a concept called co-evolution. So what you're seeing is this might be, again, this is what I was telling. These are nothing but amino acid sequence, say 50. And this might be from chicken. This might be from this row from fish. This might be from monkeys. So this might be from different organs. And that's what I mean from the row. Then you just stack them up. So this is what is multiple sequence alignment. Now you can actually see these are all same. Only this column it's different. So they are actually evolving. So that, because you cannot, when we look at a protein, whether it's fish, human, or anything, you cannot change everything. If you change everything, structure change, you lose the function. So some of the structure or some of these sequences has to stay conserved on an evolution point of view, because our body has to function. It needs that. If the structure really change, I mean, the sequence really changes, it cannot survive. So that defeats the purpose of evolution. I'm going a little bit above, but only one thing we need to know is when these are evolving together in a three-dimensional space, they would be close. So you can see this is the line. When imagine the amino acid as a bead, 50 beads. So the 50 beads like this, then they are going to form like this. So how they are going to fold in the three-dimensional space, if they're evolving, co-evolving, they'll be close. And they have to stay like this. So on a computational point of view, that is the constraint. So if you are trying to predict, this has to stay there. This cannot change. So you got a constraint you can apply for your model when you develop a model. So this is a smart way they did, because as I said, as a computational scientist, you cannot simply rely on the string and this one. You have to extract the evolution information from the biology work there. So that's what they did it here. So they actually, now in a simple way, let's think the other way. For example, if we have, let's take these first three, okay? Imagine we have the sequence for the 2, and we already know it cannot really change. So if we want to predict the third one, we know, like, okay, we know it's close relative. So it cannot change too much. For example, I'm 5'2" feet height. So my parents are 5.3, 5.4, like that. I want to have a 6 feet. I would like to have a built body, 6 feet, all those things. However, that cannot happen because my parents or my family members, if you look at the mean, it's going to be 5.2, 5.3, 5.4. So I cannot have 6 feet. So that's the constraint. That's evolutionary because I got that information. So if someone asks, hey, like if I have a nephew who's born, when he becomes 20, what would be his height? I can say it's going to be 5.3 or 5.4, but I can definitely say it's never going to be 6 feet. So that is an information you can actually extract based on the evolution, because things cannot radically change. The third output they use is the templates from protein database, which are experimental structures, which is already there. Now coming to the main part, which is our neural network. So what they did was, let me see, okay. They use this multiple sequence alignment, which has evolutionary information. So what is our idea is we have two things to combine. One, we need to extract the information from multiple sequence alignment, which is evolutionary information. Then we need the physical information, which means like positive charge, negative charge, they interact, hydrophobic. So physical properties as well as evolutionary properties. The question is, we have to use both so that we can get the structure right, because how they fold into this, it's based on this. So what they did first, they have this pairwise representation. As I said, one to say again, a chain, say 1 to 50 amino acid, 1, 2, 3. But when it comes to three-dimensional space, for the first amino acid, who would be the closest? It might be 49 amino acid. It may not be the second one. Even though sequence-wise, it's 1, 2, 3, 4, on a structural, on a three-dimensional space, it's close relative, or the amino acid which is gonna be surrounding the first amino acid might be 30 or might be 49. So that is what they're trying to determine. So pairwise information, what they do is they feed into this. So the first block is called Evoformer. It has 48 the neural blocks, so they feed this pairwise information to the evolution. So basically they are checking like, hey, is the first amino acid is close to the second? Does the evolution supports that? No. Okay, let's see. Is the third one is closest to first? Let's check. So it's keep on running like that. So once based on that, the MSA gets updated. Once the multiple sequence alignment gets updated, it just feeds that back to the pair information. So what is happening is it's a crisscross mechanism where the data exchange happens between these two domains. Now, once that is done, they do next thing. It's called triangulation. Again, they were very smart about doing this. So what they are actually trying to say is, if you know the distance in a triangle, very simple, A, B, C, if you know A to B and B to C, then this distance has a limitation. Because since we know the two sides of the triangle, the third side of the length of the third side really depends on this. So in a pairwise, for example, an amino acid, For example, this is an amino acid 1 and amino acid 49. They are coming like this in a three. If you consider as a triangle, when you need to update that, it really depends on that. They actually convert that into a graph problem as edge. So these would be serving as edge. So if you want to update one edge, it really depends on other two. So that is again another clever way for them to incorporate again the evolution as well as the geometrical constraints. So they did that. So that's a next procedure. Then what they did was, so it's a chain. They just cut into pieces. They told the structural module to interact with each other based on the physical constraints and other things. So what happened is they actually decide how they are going to fold in the three-dimensional space. So finally, after this block, which is structure, and you get a preliminary structure. Again, to improve, they actually did a concept called recycling. So you got a preliminary structure, you put that back here along with the old database, and then you are checking it again. So this recycling also significantly improved. So it has so many other components, but these are the main. So main idea. So why they got the Nobel Prize, if you ask, like they were able to incorporate all those details into this neural network. So it was not about writing those programs or running those. It was about to figure out how to incorporate the biology and physics as well as the chemicals. So they combined all those, and they were so clever to design this neural network, which was able to solve this problem. Now we got the model. How do we know they are good? So they again devised two measures. One is per residue confidence. For example, you can see the red ones, which has only the very low confidence, 50. That means the prediction is wrong. So within a structure, if you see a red region, you can say, oh, those regions are the confidence is very low. So they might not be accurate. Now, so that's about the local one. Then how about the three-dimensional arrangement? So it has multiple domains where you can see the low, the greens indicates the very low confidence. So that means, again, on a three-dimensional space, their relative position might not be accurate as the model predicted. So this is a mechanism they actually devised to say, like, I gave up new protein. Is our model prediction is correct or not? So because this is really important for us as biologists to make sure I gave up because we actually do further experiments just based on this structure. So we want to make sure it's right. Of course, then how once they got the model, they submitted for the CASP challenge, which is CASP 14. What you're seeing here is the green is experimental one and the blue is computational prediction. So what they have done is they just superimposed. So you can literally can see that they are joined to one another. So that means the prediction is good. So they use the global distance test. Basically, it says how good it is. So anything above 90 is good. So for during the challenge, their computational prediction got a score of 93 and 90, which is as good. It can be as good as an experimentally determined structure. So your prediction or your model is giving the accuracy or accurate model as good as what you get from the experiments. So that basically validates that their model is good. Now, so this is about the AlphaFold 2. This is about the it's the computational side. Now, as a biologist, how do I use it? I'm a virologist. How do I use that? And some of the things I said about the evolution, we are going to come around on the other way. So I basically work with herpes viruses, which is the viruses which causes chickenpox, STD, and it basically has a very wide host range, including humans, fish, chicken. It's basically herpes virus is so ubiquitous. It's basically infects 66% of the human population and their population. And there's a saying, it's called like, once you get the herpes, it's forever. So this is a very clever virus which I work on it. Don't worry about this slide or other details. Only the point I'm trying to make is, so this is what is the alignment. So what I'm trying to do is, these are all different virus isolates, like COVID. When we have COVID, suppose if when you go get tested as a virologist, we get those sequences. So we always ask a question, COVID, like, I don't know if when you start like Omicron variant, Delta variant, you started hearing all those, right? How do you know that? That's a fundamental question. So that's a question as a virologist I have to answer, like, okay, this is the virus which we know. Then the question comes, is this a new virus or is this a new strain, new variant? How do we do it? So simple. Again, as I said, that's where the evolution comes in. We just do the alignment and we say, okay, in this location, something happening. There is a deletion and there are some other things also happening. That means compared to what is circulating virus, which is causing COVID, there are some changes happen. So on a functional point of view, what I did for my virus, it is called channel catfish virus, which is a herpes virus. I used the AlphaFold to predict the structure. So these structures are not available. So I used the AlphaFold to do the structure. And as I said, this is an example, and this is called polymerase. This is one of the complex protein, and it's there in almost every organism. You know what its primary duty? Just make the copies of the DNA. So any organism which has the secret code as DNA, it needs this to make the copies. So it's everywhere. So I use the AlphaFold to predict the structure of my virus polymerase. Now, I told you the positive charge has an impact, right? So this is an example which really demonstrates that. So I use this is the DNA polymerase. Now there is a Coulombic electrostatic potential. What it is? It basically says how is the charge distributed on a protein. Now you can actually see the blue, basically positive. So all these regions are positively charged. These are negatively charged. Nothing fancy in there. Now, on a simple example, on a simple example, I can say the DNA. DNA is negatively charged. Now, I said polymerase has to replicate or make the copies of DNA. So that DNA actually comes through this. So it's negative charge, right? Now it's positive on the protein. So negative charged DNA coming and it's positive. So it basically brings that in. So positive and negative works. So that's the usual way. Now imagine a situation where mutation or changes happen. Evolutionarily, the things has really changed. Then what happens is, imagine a situation instead of this positive, imagine it never happens, but for our experience sake, for our argument's sake, imagine all became red, means all became negative. What is incoming? DNA, which has negative. Negative, negative. What happens? They repel. Then what happens? It cannot go through properly. It's basically interferes with the DNA replication. So it's chemistry, but that is where the evolution as well as the function plays a role. Now, for me, what happens again, that we use this a lot to track. For example, you have the COVID vaccine. It's basically spike protein. So you have a structure like this for a COVID vaccine. Now, I ask a question. So why you get a booster for a COVID vaccine every year? Because of the mutation, because virus changes. So how we design the vaccine is, we have structure like this. We usually select a region as target for vaccine. So we think when the virus has that region, but imagine virus changed a lot, this region doesn't look like that. Then what happens is our vaccines doesn't work because which was designed based on this structure. So this is a problem, and we would like to know, so the changes or mutation or evolution can happen. It happens, but there are critical areas it happens. For example, for herpes virus, if any humans, we get a herpes infection like chickenpox or an STD, you got a doctor, he's going to give you the tablets called acyclovir, which basically disrupts the structure, the virus structure. What happens if this structure get disrupted? Virus cannot replicate. So you imagine you have 100 virus in your body right now. Usually it keep on multiplying, then you get sick. But you gave the drug, it basically disrupted its DNA making machine. It cannot make its own copy. So the 100 viruses, that's it. And in a day or two, your immune system is going to go and attack it. It's going to clear it off. So if the virus wants to survive, it has to replicate so fast before the immune system catches it. It's basically hide and seek game because eventually your immune system is going to find it. It's going to destroy it. But virus, how it survives, it knows there is a time for the immune system to pick it up. By that time, it replicates so that it can create the disease. So when you give the drug, that's what it does. It basically disrupts the virus replication machinery. So virus cannot survive. So again, that is how does whether it's vaccine or drug works, it actually works like a key lock mechanism. So imagine this as the lock, the virus as a key. Now, only one on an evolutionary point, imagine the key changed. Would that work? No. So when the evolution or the new variant, new strain works, the key changed, lock sometimes remains same. Then what happens is if a vaccine or a drug has to work, it needs
Sorry, I think the resolution on the pics.
Yeah. This is the last slide. So
It's residential. Recording.
So, okay, fine. So I'm trying to conclude. So why I did put all these slides, I'm trying to give you a context why we need these structures. Because imagine coronavirus outbreak started today. We cannot wait another 3 or 4 years to determine the structure so that we can develop a vaccine or drug. So if we have the AlphaFold-like program, we can do it in 2 or 3 days. So one, as a virologist, if I know the structures and if I know what is happening, then I can develop a vaccine or a drug. And some of these things, for example, now you can actually see the amino acid changes. Let's not worry about it. I'll give you a simple example. If you know again, COVID vaccine, which is an mRNA vaccine, again, the COVID vaccine you are getting and the original virus, that has only 2 amino acid changes, 2 protein changes. Why we as scientists, so we already know the structures, we can use that as a vaccine, However, the scientists decided to change 2 amino acids compared to the original virus structure. The reason was, if you change those 2 amino acid, it gives structure, the structure more, it increases the structural integrity. which in turns can improve the immune response. So, for example, it improves the immune response for several folds. What I'm trying to say, it's only just 2 amino acids. Just 2 changes, it can change the function. So as a virologist, this really helps me to track the viruses and helps to understand how they evolve. And this was the information I was talking about. We use evolution. So some of this information is already out there. Again, the team who got the Nobel Prize, they were clever enough to get this information and put it back. So as a conclusion, what I'm trying to say is the team was made a significant effort to combine the evolution, the physical, chemical, as well as the existing template, as well as the evolutionary information to solve this problem. And this has a lot of application because now the drug design, vaccine targets, all those can be accelerated. We can solve those problems very quickly. Now, I don't know where there could be another Nobel Prize after 30 years in the same area, because there are still so many other things which has to be solved in this area. So thank you.